notes for ideas on the origin of life, which is part of the Evolutionary Theory section of biology. So first of all, I just want to say there's different ideas on the origin of life, where life came from. My job as a science teacher is to present these scientific thoughts on the origin of life. There are different thoughts, but this is the scientific thoughts. So, first of all, we know that the Earth was a very different place back when life first came about. We know that there were lots of different chemicals, there was lots of energy, lots of storms going on, lots of volcanoes. We have these things in the ocean called hydrothermal vents, where we can see the Earth is still forming and things are still happening geologically. And scientists have studied these hydrothermal vents a lot in order to try to compare that to how things might have been on the very early Earth. So we know that we have a bunch of chemicals coming out of these vents, which are chemicals that are found in living things. So scientists have tried over the years to determine how these non-living chemicals could have reacted and combined to form the first life on planet Earth, which we know is very, very simple life, single-celled organisms. Prokaryotes, remember, means they don't have a nucleus. Very, very simple bacteria. So the idea, this gradual formation of life from non-living chemicals, is called chemical evolution. And that is how scientists think that life could have evolved. So we have some scientists to learn about in this section. We have scientist Alexander Oparin. He was studying the atmosphere of the early Earth. We know that the atmosphere of the early Earth was very different than it is today. We know that there was no oxygen present in the atmosphere of the early Earth, so we would not have been able to survive back then. But they believe that the atmosphere had water vapor, ammonia, hydrogen gas, and methane. There's some evidence that has come out recently that that was not necessarily the case, but it's just really hard to know because we're talking billions of years ago, and we just kind of have to go off of the evidence found in the rocks and the things underneath in the crust of the Earth is what we're going off of. But we do know that since oxygen is so reactive, it really would not have been able to allow for living things to form because oxygen would have destroyed organic things as fast as they could be formed. We do know that there was lots of energy available for chemical reactions to take place. There was lots of storms, lots of lightning, lots of volcanoes. So possibly this energy could have allowed for chemical reactions to combine these simple chemicals to form more complex compounds. So a second scientist, J.B.S. Haldane, came up with a similar hypothesis. He also reminded us of the fact that the sun was present, so there was a lot of energy from the sun that could also cause these atmospheric gases to react to form these organic compounds which make up living things. Eventually, Oparin and Haldane started working together, combining their hypotheses and thinking about how this could have all taken place. They believe as the Earth cooled after the Big Bang, the early oceans would have formed because the water vapor in the air would have condensed, started raining, and eventually filled in the craters in the Earth, which became the oceans. And all of these chemicals ended up in the oceans in a big sort of soup, combined with this energy, which allowed them to combine and eventually evolve into organisms. So this became known as the Oparin-Haldane Hypothesis. It was an attempt to explain how life on Earth could have formed gradually from these non-living chemicals over billions of years. The reason why this is still considered a hypothesis, remember, a hypothesis has to be tested. Oparin and Haldane never had the chance to perform experiments to test their hypothesis. However, some other scientists took their idea and did set up an experiment, which is called the Miller-Urey Experiment. They set up this apparatus over here, which is a very famous scientific apparatus. This apparatus was designed to mimic the conditions on early Earth. They have the gases here. So we have your water vapor, your hydrogen, your methane gases. You have your electrode, which was designed to represent the energy in the atmosphere from either lightning, volcanoes, or the sun. We have a condenser, which allowed it to cool down and collect what was formed from this reaction that's going on in this part. And then you have a collection area over here to collect what has actually resulted from these reactions. What happened was they were able to form amino acids as a result of this. Some of you may remember that amino acids are the building blocks of proteins, which proteins are the building blocks of living things. So it was a start. They were able to take the conditions of early Earth and create compounds, chemicals that are formed in living cells and are currently part of living organisms. Another scientist wanted to take it a step further. He wanted to see if he could take the amino acids and be able to chemically combine them to form proteins. He heated up some amino acids, caused them to combine, and he ended up with things that were similar to what we've seen in current proteins, but not quite. So he called them proteinoids. Okay. And then he took the proteinoids, put them in water to see what would happen. And what happened was they ended up self-organizing into spherical membranes. And this is an actual picture up here of the result of what he got from his experiment. So these resemble cells. They look very similar to cells that we have today. They had a semi-permeable membrane, which is what cells have, which allow them to move things back and forth. They had the ability to grow and divide. So again, very, very simple cells that were similar to what we have today. They weren't exactly the same, but he ended up calling them protocells, right? Because proto is kind of like a prototype for the cells that we have now. So I'm not going to show this video on here, but eventually you'll need to click through here. And this just goes over a much better explanation of the Miller-Urey experiment than I was able to give you. So please watch that at your convenience. So our next section is on the evolution of cells. So scientists have been thinking about what the first cells were like and how they have changed over time. We know that the first cells had to be anaerobic, meaning they do not use oxygen to make energy, because we know there was no oxygen present. So they could not have used oxygen if it wasn't there. We also know that they were heterotrophic. Remember, heterotrophic means that they are unable to make their own food. So they're unable to do photosynthesis. Again, we know things were not doing photosynthesis back then, because if they were doing photosynthesis, there would have been some oxygen present in the atmosphere, because photosynthesis produces oxygen. We know they were prokaryotic, meaning they didn't have a nucleus or any other organelles. That's because things start out simple and go to more complex over time. It would not have made sense to start out with the more complex type of cell and then go to simple, because that's just not a logical thing that happens. So eventually, over time, it is believed that some of these prokaryotic cells evolved through natural selection, caused by some random mutation, to create their own food, which we now call autotrophic. There are two types of autotrophic bacteria that are still around today. We have photosynthetic bacteria that work just like plants. They use sunlight, they take carbon dioxide out of the atmosphere, and they also take water and then transform that into glucose, which they then use for energy. We also have chemosynthetic bacteria, which are found in those hot vents that I showed you a picture of earlier. They do the same thing, except instead of using sunlight, because it's not present, they use those chemicals that are coming out of the vents for energy to convert CO2 and water into glucose as well. So once those autotrophs came about and started doing photosynthesis, the byproduct of photosynthesis is oxygen. So at that point in time, there was oxygen being released into the atmosphere, which changed the atmosphere completely and eventually changed it into the atmosphere that we have today. Today's atmosphere is actually called atmosphere 3. The original atmosphere was atmosphere 1, and there was kind of a transitional atmosphere in between. But you can see in atmosphere 3, here's the oxygen, O2. We still have some water vapor. Here's carbon dioxide. Remember that the majority of our atmosphere is nitrogen gas, and then we have ozone, which is our ozone layer that protects us from the sun. So once oxygen became present in the atmosphere, we have some cells that adapted to use that oxygen to create energy. Otherwise, the oxygen, remember I said, would have destroyed everything. So the cells that were naturally adapted to be able to withstand being around oxygen and be able to then take that oxygen and use it are going to evolve into what we now call aerobic cells. So these are cells that use oxygen to produce energy, and this would be what we have. We have aerobic cells. We use oxygen to produce energy. But our cells are the result of many, many, many years of evolution changing over time to get from the bacterial cells that they had billions of years ago into what we have today. So our last thing we're going to talk about with this slides is endosymbiotic theory. This is explaining how cells went from prokaryotic cells to eukaryotic cells. So remember, eukaryotic cells have a nucleus, and they also have other organelles like mitochondria, which everyone remembers is the powerhouse of the cell. And what they believe is that mitochondria used to be a primitive bacteria on its own that was engulfed into a larger cell and formed a symbiotic relationship with that larger cell over time. The evidence for that is that mitochondria have their own DNA. There's such a thing called mitochondrial DNA. They also have their own ribosomes. Remember, ribosomes are used to make proteins. So mitochondria are like these little independent cells of their own. They also reproduce on their own separately from the larger cell that they're part of. So you can kind of see in this picture how the mitochondria got engulfed into the bigger cell, and it was an endosymbiotic relationship. So remember, a symbiotic relationship, they're both going to benefit. The mitochondria gets a home, a place to stay, and the larger cell ends up using the mitochondria to make energy. Okay, same thing with chloroplasts. Chloroplasts are very similar to mitochondria. They also have their same DNA. They also reproduce on their own. It's just kind of like the plant version of mitochondria, although plants also have mitochondria. So same thing. You have your chloroplast here, gets engulfed into a bigger cell. The bigger cell says, hey, I'll give you a place to live if you'll provide some energy for me. And that's what happened. The chloroplast was able to do photosynthesis using light from the sun, and then that larger cell benefits from that. So this is just a summary from your book on how this whole thing worked. So here is your mitochondria, which was originally its own aerobic prokaryote bacteria, okay, taking up residence inside this larger cell. Now, if it only has mitochondria, it's going to become either an animal cell, a fungi cell, or a heterotrophic protocell. If it ends up having both mitochondria and here's a chloroplast going in, it's going to become something that can do photosynthesis. So a plant cell, or some protists are plant-like and can also do photosynthesis. All right, here is an Amoeba Sisters video that explains this very well. So please watch that when you have some time to see the little animations and how this all happened. All right, and that is all that we're going to cover for today. If you have any questions, please write them down, email me, put them on Google Classroom. If you're an in-class learner, ask them in class, and I will be happy to answer them for you. So I hope this helps to clarify some things from this section of the notes. Thank you.